Hello everyone. In this lecture, we will discuss on nicotinamide adenine dinucleotide, abbreviated as NAD, an important coenzyme for the enzymes involved in the energy metabolism. Now, the lecture consists of its structure, details on structure, mechanism, biosynthesis, and the roles of NAD in the cell. NAD is a coenzyme utilized by enzymes that mainly participate in oxidoreductase reactions. It is present in all the cells because metabolism, happens in all the cells, and it is important in energy metabolism. Now, this molecule NAD is mainly important for energy metabolism because the free energy of oxidation reactions is captured in the form of electron transport potential NADH, and they finally transfer the electron to electron transport chain where it is utilized to synthesize ATP. Now what the importance of this molecule is that this molecule takes up the energy which is present in the molecules during the catabolic process and then transfer that energy into to the electron transport chain, where in the electron transport chain the energy which is there in the NADH in the form of electron transport potential will be utilized to synthesize ATP. This molecule was first identified by two British chemists, Arthur Harden and then William John Young in Yeast. Actually, it was identified as a heat stable form, a heat stable component, which is very important for the fermentation of the yeast. And then they were followed by Otto Warburg, who isolated and identified this nicotinamide moiety, and then he isolated this the whole molecule and identified the exact site where the electron hydrate transfer happens. And shortly after, Carnot Evenham demonstrated that nicotinamide, a form of vitamin B3, is vital precursor of NAD. So these are the important milestones for the discovery of this NAD coenzyme. This is the structure of NAD. We can see that this is a dinucleotide because it is made up of two nucleotides. Now, one nucleotide, there are two nucleotides here. These two nucleotides are joined by a phosphate bond here, and one nucleotide is adenine nucleotide where the nitrogen base is adenine, you can see here, and then the another nucleotide is nicotinamide. Now these two components, these are called the nucleotides because they have a nitrogen base, they have they have ribose, and then they have phosphate group. Now this molecule, to remember this molecule, we have to have to remember the structure of this adenine. Now this adenine, it is a purine base. These purine bases are made up of these are heterocycles, bicyclic heterocycles. This bicyclic heterocycle, it is formed by fusion of a 6-membered ring and then 5-membered ring, and both the rings, they have two nitrogen atoms. In one ring it is a 5-membered ring, it is in the exactly in the opposite direction, whereas in the 6-membered ring it is on the diagonally opposite direction, the nitrogen atoms. Now and then we have one NH2 group over here. This is the structure of adenine. Now then that is connected to ribose through glycosidic linkage, N-glycosidic linkage, and this nitrogen of the 5-membered ring gets connected with the carbonyl carbon atom of this ribose. Now this ribose has CH2OH at the carbon number 5. That OH group is linked to phosphate, and then that phosphate is linked to phosphate group of this ribose, and then this ribose again it is linked to nicotinamide ring. Now this NAD is nicotinamide. It is derived from vitamin B3. Now vitamin B3 has two forms. One is nicotinic acid and then nicotinamide, that we will discuss in the next slide. So now this is mainly derived from vitamin B3 because vitamin B3 is an important component here, functional component in the NAD. Deficiency of vitamin B3 leads to various symptoms and conditions which are related to energy metabolism. Now, along with NAD, I have put here NADP also. It is another very closely related coenzyme. NAD is mainly utilized in catabolic reactions where it accepts the electrons and then hydrogen atoms from the fuel molecules, whereas NADP is another coenzyme. However, it is utilized mainly in the biosynthetic pathways rather in the oxidative pathways or catabolic pathways. The main structural difference between NAD and NADP is that in case of NADP, we have here in case adenine and then ribose on the carbon number 2 of the ribose, there is just addition of a phosphate group over here. This is the only difference between NAD and NADP. Here, this NADP in the cell, it is mainly involved in catabolic reactions, whereas this one it is mainly involved in anabolic reactions. Now this NADP plus is synthesized from NAD by an enzyme called NAD kinase. Now this NAD kinase adds a phosphate group. With the phosphate group will be taken from the ATP and it will be added on to the hydroxyl group or carbon number 2 of this adenine nucleotide of this NAD to convert that into NADP plus. Now in this slide, we will discuss on how this NAD acts as an electron acceptor and then a proton acceptor in oxidoreductase reactions. Now this NAD is one of the coenzyme involved in it binds to various enzymes that participate in that actually catalyze the reactions where there is a removal of hydrogen atoms or electrons from the fuel molecules. And these there are many enzymes. For example, we have from the glycolysis we have glyceraldehyde 3-phosphate dehydrogenase. We have and then in the citric acid cycle alpha ketoglyceride dehydrogenase, and then we have malate dehydrogenase. Like that we have various enzymes which use this coenzyme NAD to take out the protons or oxidize the fuel molecules. Now all these oxidoreductases, they have a common three-dimensional structure or a moiety in their structure called Rossmann fold. This is the structure of Rossmann fold. Now to this is a signature sequence in most oxidoreductases, and this region is responsible for binding of these enzymes with the coenzyme NAD. Now this Rossmann fold consists of beta sheets and then alpha helices. They are combined in a specific sequence. You can see here these are alpha helices and beta helices. Alpha helix and beta sheets that are combined in a particular fashion to form a three-dimensional secondary structure. So this particular structure is responsible for binding of this NAD coenzyme with the enzymes. Now to this Rossmann fold, this adenine part properly binds to this Rossmann fold. This Rossmann fold will be located very near to the active site of this oxidoreductase enzymes. Now this region is responsible for making the proper orientation of this NAD so that it can properly take out the electrons from the particular parts of the fuel molecules. Now when come to the mechanism, I have taken the example of an enzyme called malate dehydrogenase. Malate dehydrogenase is an enzyme of a citric acid cycle which catalyzes the last reaction of citric acid cycle, where the malate will get converted into oxaloacetate. Here this is the structure of malate. We can see here there is an OH group which get oxidized to carbonyl CO group here. This oxidation happens with the help of this NAD. This NAD takes up the protons which are or hydrogen atoms which are present on the carbon number 2, that converts that into carbonyl oxygen. Now this if you see this, this is the structure, just to show you how what happens, what part of this molecule is responsible for taking up the electrons, and what happens when they take the electrons. Now if you see here, this is the NAD plus, which is in the oxidized state. Now this part we have, this is atom number 1, 2, 3, and then 4. Now the protons, I mean, from here will be taken in the form of hydride. Hydrides are nothing but, here I have mentioned hydride. Hydrides are nothing but an hydrogen atom with an extra electron. Now these reactions, at a time it takes the 2 electrons from the substrate molecules, and these 2 electrons will be added to this nicotine ring here. Now when the hydrogen is added here, the electrons, again it will the electrons, the double bonds here, they will shuffle, because when the electrons are first, when the hydride atom or hydride ion is added here, so this will result in breakage of this double bond, and then one carbon, there will be a valence electron carbon formation over here, and then those electrons again they will shift onto this nitrogen. Finally we will have the structure like this. Now this is how the hydrogen and electrons which are present in the substrate molecule will be taken onto this nicotine ring of this NAD. Now once it takes this NAD, and this NAD just acts as a shuttle, because these are reversible reactions, it cannot keep the electrons with itself. It has to give it to it will give those electrons to the electron transport chain, where those electron transport chain uses the electron reduction potential which is in those electrons, and that energy will be used to synthesize ATP. The same similar mechanism will happen in case of NADP plus also. In this slide, we will understand how the biosynthesis of NAD will happen. NAD biosynthesis happen by 2 pathways. One is de novo synthesis, and then another one is salvage pathway. De novo synthesis is the one where the whole molecule is synthesized from the scratch. Now the tryptophan is the important. Tryptophan is one of the amino acid or essential amino acid. Now this tryptophan will undergo degradation by a pathway called kynurenine pathway. Now in the kynurenine pathway there is an intermediate called quinolinic acid. Now this quinolinic acid forms a main precursor for the synthesis of NAD in the de novo pathway. Now in case of salvage pathway, this just utilizes already existing molecules or partly broken molecules of other metabolic pathways to synthesize the NAD. Now the cell always try to use this salvage pathway because this is less energy consuming than the de novo pathway, because the degradation of tryptophan and then to formation of quinolinic acid, it takes much amount of energy when compared to these particular reactions. Now if you see here, from the tryptophan we have quinolinic acid. The quinolinic acid, then by the action of an enzyme called quinolinic acid phosphoribosyl transferase, it will get converted into nicotinic acid mononucleotide. Now it is called nicotinic acid mononucleotide because it has a COOH group here. There is no amide group here. For this molecule, addition of phosphoribose, this is the ribose and then this is the phosphate. This particular part of this molecule, it is called phosphoribose. The addition of phosphoribose is done by this enzyme, and this phosphoribose is taken by phosphoribosyl pyrophosphate. Now, nicotinic acid mononucleotide can also be formed from a nicotinic acid. As I told you, nicotinic acid is one of the form of vitamin B3. We have nicotinamide. This is another variant or type of this vitamin B3. Now, nicotinic acid, by the action of enzyme called nicotinic acid phosphoribosyl transferase, there will be addition of this ribose and phosphate group, and that ribose and phosphate will be taken from the ATP. Finally, this nicotinic acid will get converted into nicotinic acid mononucleotide. Now, this nicotinic acid mononucleotide, first thing is that it has to be added with an adenine group to convert that into NAD, and it also needs to be here, this COH group should be converted into CONH2, then only it can become NAD. Now, to complete that reaction, nicotinic acid mononucleotide, by the action of enzyme called nicotinic acid mononucleotide adenine transferase, there will be transfer of adenine group. You can see here there is a transfer of addition of an adenine group over here. Now, when the adenine group is added here, now that adenine group will be taken from ATP, and the APPA will be eliminated out here. Now, when the adenine group is added, this will become nicotinic acid adenine dinucleotide, NAAD. It is not yet NAD because it has here a carboxylic group here. Now, nicotinic acid dinucleotide, by the action of enzyme NAD synthase. Now, this NAD synthase is an enzyme, is a transaminase enzyme. It takes up the NH2 group either from NH3 or glutamine and another enzyme, another amino acid, NH2 containing amino acid in its side chain. Now, that amine group will be taken by this enzyme and it will be transported onto here to synthesize an amide group over here. Now, when that happens, now for this it requires much amount of energy. That energy will be utilized, that will be generated from hydrolysis of this ATP into AMP and PPI. Now, finally we have nicotinamide dinucleotide or NAD, the synthesized NAD here. Now, this is a de novo pathway where quinonic acid, nicotinic acid mononucleotide, and then nicotinic acid dinucleotide, and then finally into nicotinamide. Now, the salvage pathway from the nicotinic acid, again it also uses the same pathway, but the substrate is not quinolinic acid but nicotinic acid, which is a form of vitamin B3. Now, these two pathways are called deamidated pathway because these groups here in this nicotine part, they do not contain CONH2. That is why they are called deamidated pathway. Now, in a salvage pathway, we have two other ways in which the NAD can be generated. One is by nicotinamide. You can see this is the difference between nicotinic acid and nicotinamide is here. COH group is there, CONH amide functional group is here. Now, nicotinic acid, nicotinamide, by the action of enzyme called nicotinamide phosphoribosyl transferase, there will be addition of a ribose and phosphate group at this particular position here to convert into nicotinamide mononucleotide. Now, this nicotinamide mononucleotide can also be synthesized from nicotinamide riboside. Now, it is also a form of vitamin B3. Now, nicotinamide riboside, by the action of enzyme nicotinamide riboside kinase ATP, now here it will get converted into nicotinamide mononucleotide. Now, this nicotinamide mononucleotide, by the action of enzyme nicotinamide mononucleotide adenine transferase, it will get converted into NAD. Now, this is the different pathways in which the NAD is synthesized in the cells. Now, in this slide, we will discuss on roles of NAD in cells. Now, already we have discussed, we have understood what are the important roles of NAD. NAD, as I told you, it involved in the oxidation reduction reactions or catabolic reactions, or oxidoreductase enzymes will utilize them, this NAD as their coenzyme part. Now glycolysis, it is utilized in glycolysis, it is utilized in conversion of pyruvate to acetyl coenzyme A. Now we know that glucose will get converted into two molecules of pyruvate, and then by the process of glycolysis, and then pyruvate will get converted into acetyl coenzyme A by the action of pyruvate dehydrogenase. And then we have a TCA cycle, we have isocitrate dehydrogenase, we have alpha ketoglutarate dehydrogenase, and then malate dehydrogenase that will generate NADH. Now where, I mean, in these steps NAD is used as a coenzyme. Now NADH is an now whatever the NADH, I mean whatever the electrons which are taken up by this NAD from all these metabolic pathways to get converted into NADH, and then finally it will move to electron transport chain, where this there is a process called oxidative phosphorylation. It is called oxidative phosphorylation because the energy which is captured in by the process of oxidation by this NADH will be converted into will be utilized to phosphorylate ADP into ADP to ATP. Now this is the main important thing that is, I mean, important function that is carried out by the NAD. And then along with serving as important component of oxidoreduction, I mean energy metabolism, they are also part of various regulatory enzymes such as mainly the sertolins. Sertolins utilize NAD because they are NAD dependent deacetylases. These deacetylase they mainly deacetylate the acetyl group which are present on the proteins such as histones or any other regulatory proteins, and thus they serve as a regulatory agents. Now for these sertolins to be active, they require NAD plus. Now the another regulatory proteins, they are called PARPs. These PARPs, they are called adenosine ribosyl polymerases. Now these PARPs, what they do is that they will add phosphorase adenosyl groups onto various proteins, and they participate mainly in the DNA damage response, because wherever the DNA damage is there, now this part will go and bind, and then what they will do is that they will take phosphoribosyl adenosine phosphoribosyl group from the NAD, and then they will add that onto the various different proteins to activate them and also to recruit DNA damage repair systems. Now because they exclusively utilize NAD, if the NAD serves as a regulatory component for the activation and functioning of this PARPs. Now here also there is a system called CADPR and then NADP, where this NAD derived messengers that regulate intracellular calcium signaling. And also very importantly, NAD and NADH ratio, it is very important because many of the enzymes of glycolysis, many of the enzymes of citric acid cycle, they are regulated by the concentration of NAD or NADH, because this NAD, it acts as an stimulator for various catabolic enzymes. So because of that, the ratio of NAD and NADH serves as a key component to either activate or to reduce the activity of various catabolic and anabolic pathways. So this is the summary of my presentation. We have understood that NAD is a dinucleotide. It is a coenzyme existing in NAD and NADH, and another very closely relative coenzyme NADP plus and then NADPH. Now the core chemistry, it is the mechanism of this coenzyme to transfer the electrons is a type of reversible hydrate transfer at the nicotinamides rings at C4 position. This is the central hub for catabolic energy metabolism because it is involved in various, it is a main component of enzymes involved in glycolysis, TCA cycle, and oxidative phosphorylation. Along with participating as energy metabolism, they also are the substrate for regulatory enzymes such as sirtuins, PARPs, and then calcium signaling molecules synthesis. And then the NAD levels link cellular metabolism to gene regulation, DNA repair, and aging. Now it is postulated that with age, the concentration of NAD will start to come down. Because of that will negatively affect the metabolism and thus increasing the aging and senescence process. Now this completes this particular presentation. Thank you.